There is still a lot of discussion about PD-L1 testing in non-small cell lung cancer, and many of the discussions centre around whether we need to test or not. We can do away with testing, but most of the questions centre on the issue of how we should carry out the test. And the reason for this controversy is that there are now a number of drugs on the market or coming to market, with each with their own developed PD-L1 biomarker test. However, it is not feasible in the vast majority of pathology laboratories that we can offer 5 or 4 or even 2 different tests. The ideal scenario would be for us to be able to offer one PD-L1 test, which can be the data from which can be used for any of the drugs in any of the lines of treatment that the oncologist wishes to use that biomarker information. So the challenge for the pathology community has been to try to figure out, are the trial validated tests equally effective at selecting more or less the same group of patients, and when those patients are selective, are they going to benefit from the therapy in the same way as they would had they been selected in precisely the way that was carried out in the trials? So we have a variable amount of data to try and address some of these questions. We have quite a lot of data now from a number of different comparative studies showing us that of the 5 trial-validated assays that are out there, 3 of those assays are relatively similar, or similar enough that they are probably interchangeable in terms of clinical use for selecting patients for biomarker therapy. Those assays are the assays based on the 28.8 clone, the 22C3 clone, and the SP263 clone. I think in most people's regular practice, if they are using a trial-validated assay, they're probably using, I don't have hard data, but they're probably using either the 22C3 assay or the SP263 assay. So from a technical point of view, these assays are interchangeable. The big question has always been, will the clinical outcomes be the same? And we've had very little data to work with so far. But recently there was a paper published which suggested, at least in second line for treatment with nivolumab, and I would remind you that we do not absolutely require in most countries testing for second-line nivolumab, but nonetheless, this study was carried out in Japan, and it demonstrated that these assays were interchangeable in terms of the same clinical outcome being determined according to selection by these different assays. So that's encouraging data, but we need more of it.